Hi guys, my name is Ali Pezzalezzi and I will be presenting figure 4 today for our research article. And in my figure there are 3 parts, A, B, and C, so I want to just dive into what each of those represents. So figure 4A represents the role of PAM, which is a protospacer adjacent motif in target DNA cleavage by the Cas9 track RNA cRNA complex and the CRISPR-Cas system's recognition of self versus non-self. The role of PAM in target DNA cleavage, we found that it was particularly sensitive to one of the strands of DNA. And in the CRISPR-Cas system, the recognition of self versus non-self involves a short sequence in PAM, which is essential for protospacer plasmid DNA elimination. Figure 4B represents the use of cRNA sp4, which was not used in the previous figure 4A, and protospacer DNA. And we selected this due to the use of its colonylical PAM, which was not present in figure 4A, and that we use this in Cas9 catalyzed DNA cleavage. Figure 4C, we looked at the role of PAM and how it plays a direct role in recruiting the complex to the correct site. And this shows a mutation that affects the binding affinity of the Cas9 track RNA cRNA complex for the target DNA. So we're going to go into the methods. They used a DNA cleavage assay in figure 4A. So they began with a short DNA oligonucleotide, and they radiolabeled both strands of the double-stranded DNA on the 5' end. In this particular figure, it was cut at 50 NT and 23 NT. The next one I think we're all pretty familiar with. This is gel electrophoresis, and you're first going to get your glass plate and put masking tape up on the sides, and then you're going to pour your agarose solution onto the glass plate, put that comb, push it down into the gel to form your wells, which we're all pretty aware of. You're then going to take the DNA segments that you're using, that you're testing, and you're going to load it into your micropipette, and you're going to use that micropipette and pipette it into the wells very carefully, if I may add. And once you do that, you should see DNA fragments move through the gel toward the positive electrode, and that will give you your results. The third method that we used are gel mobility assays, and this was used in figure 4C. So this was used in determining the role of PAM and if it recruits the Cas9 track RNA cRNA complex to the correct target DNA site. So you're going to do this by preparing your DNA by purifying it, and then preparing your complex, your Cas9 track RNA cRNA complex, whatever you're putting into the DNA. And then you're going to use non-denaturing gel electrophoresis to ensure that none of your DNA gets denatured, and you should be able to detect specific DNA sequences. In this case, you'll be able to see if our complex was able to target the correct DNA sequences. So these are the results. This is figure 4A, and in figure 4A to the right, we can see the CRISPR-Cas system and its recognition of self versus non-self. And like I said in the beginning, there's a specific motif within the PAM sequence that is necessary for this protospacer plasmid DNA elimination by the CRISPR-Cas in only bacterial cells. So as you can see, this GG is the necessary sequence, and it's located one base pair downstream of the cRNA binding sequence within the target DNA. So when we looked at transformation assays, they demonstrated that this GG motif is the essential motif for this plasmid DNA elimination. So you can see here, there's a GG, and here there aren't. So you can see that the complementary strand was not touched because there is no corresponding GG to the CCs. But in this, you can see that there are GGs. And then to the right, we're looking at the role of PAM in target DNA cleavage by the complex. And like I said, we tested that in a series of double-stranded DNA duplexes that contain mutations in their PAM sequences. So we were looking at the complementary and non-complementary strands, or both. And the cleavage assays showed that the DNA cleavage was particularly sensitive to mutations in the PAM sequence, but only on the non-complementary strand of DNA. So we have 50 NT and 30 NT, and you can see that in 5, 6, 7, and 8 that they are darker and that they don't really show up in 30 NT. So you can see that there's certain the PAM recognition of the complex recognition of mutations in the PAM sequence only really happened in the non-complementary strand versus the complementary strand, where in the type 1 CRISPR-Cas system, that is where DNA cleavage is most effective. In our figure B, I first wanted to point out that you can tell that the non-complementary DNA strand is cleaved at one or more sites because it appears at PAM 1 and 2 and in our control, obviously, whereas the complementary strand is not. So you can see that the non-complementary strand is first cleaved nucleotically. I can't say that word, sorry. And then it's trimmed 3' to 5' end by exonuclease activity. In this particular figure, we were looking at the use of the crRNA sp4 and protospacer 4 DNA, which was not used in our previous figure 4A. And like I said, this was selected due to the presence of the canonical PAM, which was not present in protospacer 2 target DNA. And I was mentioning before that there's a specific nucleotide that is necessary for efficient Cas9 DNA cleavage. And in this figure, you can see that it is G. They found that both G nucleotides were required for efficient Cas9 catalyzed DNA cleavage. So here you can see that it's only present in the non-complementary strand, which appears about 3 to 5 base pairs downstream of, I think, the target sequence. And then in figure 4C, you can see the role that PAM plays in recruiting the Cas9 track rcRNA complex to the correct DNA site. So they used gel mobility shift assays, which is what you see here, and is what I talked about in the methods. And you can see that in the unbound right here, it appears in PAM1 and in PAM2, but in the bound there are barely present. So to dive further deep into that, they analyzed these binding affinities of the target DNA sequences, and they found that mutations of either the G in the PAM sequence substantially reduce the affinity of the Cas9 track RNA-cRNA complex for the target DNA. So when it's bound, when the G is bound, you can see that the complex does not bind to the correct sequence. And when it is unbound, you can see that it binds to the target DNA sequence. So that is figure 4C unfolded for us. And diving into the conclusions, like I had mentioned, in figure 4A, the GG motif is the essential motif in the protospacer adjacent motif, and that is essential for that protospacer plasmid DNA elimination in bacterial cells by that CRISPR-Cas system. And that motif is also essential for in vitro protospacer plasmid cleavage by the track RNA-cRNA guided Cas9. DNA cleavage was also particularly sensitive to the mutations in the PAM sequence on the non-complementary strand, but like I said, in type 1 CRISPR-Cas systems, it is the complementary strand. In figure 4B, we had found that the G nucleotides of the PAM were required for Cas9 catalyzed DNA cleavage, but we also found that in figure 4C that the mutation of those Gs in the PAM sequence actually reduces the affinity of Cas9 Trac9C RNA for the target sequences. I just wanted to say that this finding argues for specific recognition of the PAM sequence by Cas9 as a prerequisite for target DNA binding. And this also possibly allows for strand separation, for strand invasion, for R loop formation, which would be analogous to the PAM sequence recognition by that Cas A.